Cell division is a very important process in all living organisms. During cell division, cell growth also takes place. DNA replication occurs before the cell divides. Cell growth, DNA replication, and division occur in a coordinated manner. The parent cell divides to form two daughter cells containing intact genomes. A typical eukaryotic cell cycle can be observed in human cells growing in culture. Human cells grown in culture show the different phases of the cell cycle clearly. Human cells grown in culture show the different phases of the cell cycle clearly. A typical human cell completes one cell cycle in approximately 24 hours. During the G1 phase, the cell grows and prepares for DNA replication.
In the S phase, DNA synthesis takes place and chromosomes are replicated. During G2 phase, the cell prepares for mitosis and checks replicated DNA.
Mitosis is the most dramatic period of the cell cycle. Prophase is the first stage of mitosis and follows the S and G2 phases of interphase.
During the S and G2 phases, newly formed DNA molecules remain intertwined and indistinct.
Prophase begins with the condensation of chromosomal material. During chromatin condensation, the chromosomal material becomes untangled.
The centrioles duplicate during the S phase of interphase.
The duplicated centrioles begin moving toward opposite poles of the cell. The complete disintegration of the nuclear envelope marks the beginning of metaphase. As the nuclear envelope disappears, chromosomes spread throughout the cytoplasm. By metaphase, chromosome condensation is fully completed. Anaphase begins immediately after chromosomes align at the metaphase plate. The sister chromatids separate and become chromosomes of future daughter nuclei. The separated chromosomes begin migrating toward opposite poles. A key event of anaphase is the splitting of centromeres. The daughter chromosomes continue moving toward opposite poles of the cell.
Telophase is the final stage of mitosis and begins after chromosomes reach opposite poles. During telophase, chromosomes begin to decondense and lose their compact form.
The Golgi complex and endoplasmic reticulum reform during telophase. Telophase ends with the complete formation of two daughter nuclei. After karyokinesis, the cell itself divides through a process called cytokinesis. Cytokinesis divides the cytoplasm and completes cell division. In animal cells, cytokinesis begins with the formation of a cleavage furrow.
The furrow finally joins at the center, producing two daughter cells.
In some organisms, cytokinesis does not occur, resulting in a multinucleate syncytium. Mitosis is also known as equational division because chromosome number remains unchanged.
Mitosis usually occurs in diploid cells and produces diploid daughter cells.
In some lower plants and social insects, even haploid cells divide by mitosis. Mitosis produces daughter cells with identical genetic information. Mitosis plays an important role in the repair and replacement of damaged cells. Mitosis is essential for growth, repair, maintenance, and continuity of life.
Meiosis is a special type of cell division involved in sexual reproduction.
Sexual reproduction begins with the fusion of two haploid gametes.
Only one cycle of DNA replication occurs before the two meiotic divisions.
A key feature of meiosis is the pairing of homologous chromosomes. At the end of meiosis II, 4 haploid daughter cells are formed.
Meiosis maintains chromosome number and creates genetic variation in sexually reproducing organisms. Prophase 1 is the longest and most complex stage of meiosis.
During leptotene, chromosomes gradually become visible under the microscope.
Chromosome condensation continues throughout leptotene.
During zygotene, homologous chromosomes begin pairing together.
The pairing of homologous chromosomes is called synapsis.
The paired homologous chromosomes form structures called bivalents or tetrads.
Crossing over exchanges genetic material between homologous chromosomes.
The enzyme recombinase controls the process of crossing over.
By the end of diakinesis, the nucleolus disappears and the nuclear envelope breaks down.
Diakinesis marks the transition from prophase 1 to metaphase 1.
According to cell theory, new cells arise only from pre-existing cells. Cell division continues throughout the life cycle of an organism. Meiosis is a special type of cell division in sexually reproducing organisms. During meiosis, the chromosome number is reduced by half.